Hello everyone, this is Byung-Chot at ExoCoBio. Today I'd like to share two scientific findings. The first one is a new publication on the adipose stem cell exosomes for ultimate reverse aging. And the other is that the IL-6 reduction and anti-inflammation mediated by adipose stem cell exosome, which is the same as ExoSCRT exosomes. In 2022, I have been speakers for more than 15 aesthetic or anti-aging conference, including IMCAS, Dubai Doma, AMWC, FACE, MEDAM, and others. I'm very proud to be speakers for all of these conferences, and I will be speakers for uh, much more conferences like these. Here, this is the key concept of ExoSCRT exosome, which is one kind of adipose stem cell derived exosomes. We believe that there are three main functions of our exosomes. The first one is anti-aging, the second is rejuvenation, the third is the anti-inflammatory function. All these three functions can create synergistic effects to keep your skin or hairs or scalp young and healthy. So this is the very brand new research article which came out in last October. Let's you get the title. As you can see here, the small extracellular vesicles from young adipose stem cells can prevent frailty, improve health span, and even decrease epigenetic age in old mice. This publication was done by Dr. Consuleo Borras. She's living in Valencia, in Spain, and she and her team proposed that young, small extracellular vesicle treatment can promote healthy aging. Actually, last December, I visited Dr. Consuelo Borras to listen to her brilliant scientific finding on this publication. And the visit to her was a great experience actually. And here in this slide, you can see the overall protocol and the key results, which I'd like to share today. So in panel A, you can see that on day 1, adipose stem cell exosomes were injected intravenously and 2 times for 30 days. And at day 30, they sacrificed their mice, all the mice, which are on average 22 months old. And after sacrifice, they checked the total body weight, which was not statistically significant between groups. But when they checked the grip strength, which is a key measurement from this study, the group of small extracellular vesicle, adipose stem cell exosome treated group, showed much more grip strength. And they checked fatigue resistance, and most importantly, this is very interesting, the frailty score defined by their collaborators at day 30, they couldn't see any frailty score from the adipose stem cell exosome treated group, which is very interesting and surprising and very definitive proof that adipose stem cell exosome can reverse aging from these mice. Also, here you can see that in panel G, when they checked the hair regrowth, they could see that adipose stem cell exosome showed almost 2.5 times more hairs at day 14. In this panel H, you can see and you can compare the hair growth from the beginning and day 14, and the hair on these mice. This mouse showed the completely recovered hair on their back. And there was no difference between male and female. It's quite interesting. So adipose stem cell exosome can work for both males and female mice too. And when they measure the urea concentration, the difference between groups, they found that adipose stem cell exosome treated group showed lower plasmatic urea, which means that their kidney are healthier than the control group of PBS. And what I liked most from this study is that small extracellular vesicle from young adipose stem cells can ameliorate molecular trait of aging in the kidney and muscle as well. And they showed lower senescence in old mice. And the key data here, what I focus on is that IL-6. So everybody says that IL-6 is a key factor of cellular senescence, and it's a key factor of SASP. And in every panel, you can see that statistically significant data or not. It depends on what you try to measure, but definitely in terms of inflammation or cellular senescence, in panel E, you can see that the IL-6 concentration was lowered or reduced after in the group of adipose stem cell exosome treatment. And this was almost the same in kidney as well as muscle, which means that adipose stem cell exosome can work globally or systemically. And much similar uh, in IL-1 beta concentration too. So there have been a lot of similar animal studies or in vitro studies from this experiment, but I believe this publication is the most surprising and the greatest anti-aging experiment, which proves the ultimate reverse aging function of adipose stem cells. And this is the second part I prepared today, and I have been interested in the role of IL-6 in skin or inflammation in skin. And you can find this article that IL-6 is related to fibrotic disease. And this in summary, IL-6 can be a inflammatory product to stimulate JAK-STAT signal pathway, and then they can initiate fibrotic process in skin, eye, lung, even in kidney. So a lot of researchers are trying to find the key of these kind of fibrotic or inflammatory process. And I found another interesting article here that IL-6 can be a new therapeutic agent or target, I mean, in systemic sclerosis, which is very rare and hard to cure autoimmune disease. So in this abstract, you can see that IL-6 is key to the inflammation or fibrotic conditions of systemic sclerosis. So here you can see that elevated levels of IL-6 are found in systemic sclerosis patients, and this correlates with skin sickness. And so a few anti-IL-6 antibodies are being developed to treat this kind of serious disease. But I believe that adipose stem cell exosome, like ExoSCRT exosomes, can be a new modality or can be new class of molecules, very complicated molecules to treat these kind of uncurable diseases. So this is one of our internal data, and some part of this data already published. So we use this kind of in vitro assay model sometimes, and then LPS treated macrophages are producing IL-6 or other inflammatory cytokines. But when we add our exosomes, adipose stem cell exosomes, it can reduce the production level of IL-6. As you can see here, it's a dose-dependent manner sometimes, and then it's a very strong or potent when we compare the potential of ASCE and the dexamethasone, which is a kind of steroid. And on the right side, you can see that IL-27 interleukin-1 beta were also suppressed by the treatment of adipose stem cell exosomes. So in the publication, as you mentioned, in the publication of Dr. Consuleo Borras, IL-6 can be a key mediator of aging or inflammation, and they can be successfully suppressed by the treatment of adipose stem cell exosomes or ExoSCRT exosomes. Here, I'd like to mention briefly the second publication done by ExoCoBio, which is about the skin barrier reconstruction and lipid metabolism, each all related to IL-6 too. So in this animal model and this publication, we found that adipose stem cell exosome can induce de novo synthesis of ceramide to re-epithelialize or to strengthen skin barriers, and they can induce the production of skin barrier promoters. And the total ceramide or total dihydroceramide, which are key lipids in our skin barrier, were upregulated, while dexamethasone, the steroid, couldn't. And one figure in our publication, you can see that IL-4 and other important pro-inflammatory cytokines were downregulated, and TSLP is a key itching molecule, was also repressed by adipose stem cell exosomes. So, based on these scientific discoveries, we believe that aging is the loss of adipose stem cells and the loss of adipose stem cell exosomes. So, we are trying to provide the lost or compensate for the loss of adipose stem cell exosome, in addition to other important ingredients to create skin regeneration or to reduce cellular senescence too. So in this slide, you can compare the level of re-epithelialization of skin of incision model of pig skin. So after incision, we applied our finished product and our API, adipose stem cell exosomes, topically. So after 19 days, we could see very successful re-epithelialization for both groups. And this is kind of old data, but I'd like to share this because we applied our API or products topically, and then they worked well to stop dermatitis, and they can create re-epithelialization successfully, which are very helpful for this dermatitis patient. And last year, we got another case from Mexico, Dr. Jose Manuel Pino. And you can see here, this patient, dermatitis patient, also have benefited a lot from the treatment of our products. His dermatitis and his skin condition was recovered to improve his quality of life before and after. I thank you and I appreciate this result of Dr. Jose Manuel Pino and Obernia. And this is another case which we got last year, and Dr. Shamio Danni, our UK KOL, generated this case study. And this lady suffered from a serious side effect of inflammation after Pfizer mRNA COVID-19 vaccination. And she could successfully improve her skin condition. As you can see here, it's quite surprising that it's much better than we could expect last year actually. And we believe that our product is working. And this is my maybe last slide. And we had a great event about 3 months ago, RAES, which means the Regenerative Aesthetic Exosome Summit. And we had about 200 people, including our doctors from the US, UK, and France, and many from many countries. And we had a lot of interesting talks, seminars, and events from our brilliant exosome scientists, including Dr. Jan Rotterbar and Dr. Sakyang Lim, and our key doctors and dermatologists from the world. Thank you for your listening for my presentation today. And you are seeing that our brand new the ExoGMP facility which is the largest stem cell exosome production facility in the world. I hope some of you guys could join the second RAES event next year to look around this brand new and top tier facility for your practice and for your scientific knowledge. Thank you.